Every year, millions of people are diagnosed with cancer. The scientific community is continuously striving to find better ways to diagnose and treat cancer. Nuclear medicine makes use of the intrinsic radioactive decay of several radioactive atoms, coupled to drug molecules which are highly specific and effective to diagnose or combat cancer. Current research in nuclear medicine builds further on an already well-established and scientifically recognized base, but there is still a huge potential to be exploited, and breakthroughs are discovered daily. Let's take a journey into the translational research of how we can get to a radiopharmaceutical for cancer treatment. A good understanding of cancer biology is essential before radiopharmaceuticals can be developed and evaluated. As the name suggests, a radiopharmaceutical is both a pharmaceutical and radioactive, where the pharmaceutical guides the radionuclide to the cancer. Based on the decay properties of the radionuclide, these radiopharmaceuticals can be used to diagnose or treat the cancer. Research nowadays is aiming to combine both therapeutics and diagnostics in what is called the theranostic approach. Here, the pharmaceutical molecule is the same, but the radionuclide can be changed based on the needs, therapeutic or diagnostics. This allows for specific and effective targeting of the cancer. Radioactive elements used in nuclear medicine are not occurring naturally and have to be generated in a nuclear reactor or via accelerators, such as a cyclotron. Once a potential candidate pharmaceutical is found in combination with the right radioisotope, various approaches in radiochemistry are applied to efficiently label, that is, couple the pharmaceutical and radionuclide, the radiopharmaceutical. This radiopharmaceutical will then be preclinically evaluated. Typical tests include binding assays, functional assays, autoradiography studies, and stability studies. Here, radiobiological properties can be determined, including the efficiency of the radiopharmaceutical to damage cancer cells and what the toxic effects on healthy tissue are. Next, in vivo tests will be performed to determine how the radiopharmaceutical behaves in a living body. In these tests, certain pharmaceutical parameters such as the distribution, metabolization, and excretion are studied. Similar research can then be conducted on tumor-bearing animals, which also includes studies to determine the efficiency to stop or even reverse tumor growth. Before a radiopharmaceutical can move to the clinical stage, the acquired knowledge has to be combined with data on dosimetry to get the right dose tailored to the characteristics of the patient. The dosimetric response and toxicity are determined on a cellular level, tissue level, afterwards on patient level. Furthermore, different aspects must be checked with regard to the safety of the radiopharmaceutical before it can be administered to a patient. Quality control and standardization of all these techniques are crucial to ensure good laboratory practice for these tests in vitro and in vivo. Finding the optimal candidate, radiopharmaceutical does not stop in the lab. In order to enable worldwide access to hospitals, the whole production process must be industrialized and optimized. Typically, radioisotopes can be produced via neutron irradiations in nuclear research reactors or particle accelerators, where protons or other particles are used to convert a non-radioactive material into a desired radioisotope. Recently, an alternative method to produce high-purity radioisotope samples is by irradiation, extraction, and mass purification online. This is called an ISOL system. Radioisotope production facilities must be designed in such a way that they can produce large quantities in an economical, efficient, and safe way. The extraction and manipulation of radioactive materials is done in dedicated installations, warranting a high degree of safety. Reactor-based facilities, as well as accelerator-based facilities, are not commonly available. Many challenges exist in the design and operation, which require a highly specialized team of scientists and engineers. From the moment of production, the radioisotopes start decaying, which means that the initially created amount of radioactivity decreases as a function of time. This means that every next step is a race against the clock to get the radiopharmaceutical in a ready-to-use format to the patient. The manufacturing process of a radiopharmaceutical typically contains several steps. The first steps of this industrial process involves radiochemical purification and separation of the irradiated material coming from the reactor or accelerator to extract exactly the needed radioisotope. One of the most effective ways to physically separate isotopes is based on the mass difference of the radioisotope. This can be done in an isol system operated without the accelerator beam. In the following step, the radioisotope is coupled in a chemical process with a given molecule designed to target specific cancer cells in the body. To ensure a high degree of safety for the patients, radiopharmaceuticals need to be manufactured under GMP, a quality management system that guarantees that the production occurs according to the highest standards and that all steps are documented. Before releasing the radiopharmaceutical for human use, a thorough quality control of the product is performed. Only in this way can it be made sure that the product does not contain any impurities that will harm the patient. Translational research enables the pathway of discovering and developing good radioisotopes, which might lead to excellent diagnostic and therapeutic means to fight cancer. This requires a multidisciplinary approach between scientists and professionals in service of the medical community.